And yeah, take it away.
Here we go. Yeah, thank you so much. Really appreciate the opportunity to be here and to share some thoughts with you. What I'm going to do today is, when I got the description of the department and how wide the interests here are, I try to sort of put something for everyone. So this talk has a mix of things, which could work well or could go badly. So we'll see. But there are some very specific things in terms of biomedicine and how we try to approach regenerative kinds of therapeutics and so on. But at the same time, I tried to really cover some pretty philosophical ideas, some very basic things on the nature of cognition and things I've been working on as far as understanding what selves are and how minds scale from the simplest organisms. And to me, it's really important that these two things are tightly connected. So in my group, any of the conceptual frameworks that we make are meant to drive new science forward, and that's how we think we know we are on the right track or not, by seeing what kind of discoveries, including therapeutics, these things are able to push forward. And we like to take the findings that we have and use them to sharpen our philosophical intuition. So we'll see to what extent that's possible. So anybody that's interested in the primary data, the software, all the original figures, everything can be found at this website. So please don't hesitate to contact me if you want to ask questions later or download things and so on. So let's just start with this. This is a classic, very classic painting called Adam is Naming the Animals in the Garden of Eden. And the idea behind this back in the day was that names are very powerful. Names were supposed to reveal the true nature of something. And in particular, names for specific things suggest crisp categories. And that's one of the things I want to try and blow up today is this idea that there are these particular natural kinds that we have to keep fixed. And so you can start out this way, and many people do that. We've got this idea of a human, a standard human, and that standard human has a sort of an agential glow about him that doesn't really extend very well to the rest of nature, and that when you try to understand where these complex cognitive capacities come from, oftentimes people say things like, well, that's anthropomorphizing, meaning that you're taking something that is magical, it's only in humans, and it's really inappropriate to see it in other places. But if we take evolution and developmental biology seriously, we find out that, well, this human is actually a continuum of selves. First of all, certainly on an evolutionary time scale, right? So there were these humans and then some other types of humans and some stuff before that. But even at the lifetime, within the scale of the lifetime of this individual, we come from a series of transformations. And there are other places where these transformations are very interesting in the world. For example, here is a caterpillar. This soft-bodied creature lives in a two-dimensional world. It eats leaves. It has a brain that's suitable for that kind of purpose. Eventually, it has to transform into this completely different creature that has to live in a three-dimensional world. It flies, it drinks nectar, it doesn't care about the leaves anymore. And so during this process, what happens is that the brain is basically dissolved. Most of the connections are broken. Many of the cells are killed off by apoptosis, and a new brain is rebuilt. A couple of interesting things. One is that there's pretty good evidence that memories survive this process. So if you train the caterpillar on something, the moth or butterfly will remember the original information. That's despite the fact that the memory medium, supposedly, the brain, is being thoroughly sort of trashed and redone. The other thing is that this has interesting sort of philosophical implications, because as you think about what's it like to be a butterfly, you can take that question one step further and you ask, well, what's it like to be a caterpillar slowly changing into a butterfly? And that makes the changes that we undergo, let's say during puberty or so on, make them seem rather minor. But in any case, we really need to understand what it means to remain the same, and for example, using a continuity of memories here, and what it means to change, and what happens to our self during that process. It's actually even worse than this. This is already sort of disturbing enough, but it's actually worse than that, because you can go lateral. And we now know that using synthetic biology, bioengineering, at every level of biology, we can substitute various kinds of engineered or in fact biological other systems. And so we can make all sorts of weird hybrids and of course instrumentize and I have all kinds of extensions, and so there's a continuum not only here, but there's a continuum of all kinds of possible beings here. And so what we find out is that while many of our categories are sort of discrete, the actual underlying biology is quite continuous. All these changes can be made extremely small, and we need frameworks to deal with all of this. So I've been working on this idea of creating a framework that would allow us to think about simultaneously not just familiar kinds of intelligences. So that would be humans, other mammals, birds, maybe an octopus. We need to have a framework that handles everything. So all kinds of weird creatures, colonial organisms, swarms, new synthetic life forms that don't exist yet, but are going to. So all kinds of strange bioengineered things, software intelligences, and someday perhaps even exobiological agents. So we need to understand, what are the invariants? What do all of these things have in common? I think it's completely implausible to think that everywhere in the universe, the only thing that has true cognition must have a brain that looks like ours and a frontal cortex and all that. So we need to go beyond that, and we need to understand what do all these different embodiments of agency have in common. So this, of course, is not the first time people have recognized the need for that. So here in the 1940s, Rosenbluth, Wiener, and Bigelow already started on a cybernetic framework, starting with very simple passive kinds of Bratenberg vehicles and very simple kind of agents, and try to lay out a map of some of the great transitions, but really understanding that these are all of the beings, from the simplest to the most complex, with human metacognition and whatever happens to be beyond. They're all part of one spectrum, and there are certainly steps on that spectrum, but they're all part of one spectrum. And the other nice thing about this kind of framework is that it doesn't say anything about brains, and it doesn't say anything about whether something was evolved or designed. It's very functional. I think this is the right way to think about it. So because I'm really interested in kinds of conceptual frameworks that help research directly, I tried to come up with a scheme that wasn't really wasn't founded on the philosophical aspects, but really from a very engineering perspective, right? And so what I have here is something I call an axis of persuadability, and these are just 4 sort of waypoints on that axis. There are many more, but what it tries to do is it tries to tell you that whatever system you're looking at, whether it be living or non-living or whatever it might be, the real question is, how do we relate to it? Meaning that what is the most efficient way to make predictions and make changes in the function of that system, and specifically to insist that that is an empirical question. You can't sort of sit in your philosophical armchair and say, I don't believe this thing has any intelligence, or it does, or whatever. You have to make a model. You have to choose what kind of tool you're going to use along the spectrum of persuadability. And then we have to see, and we have to check and see if that actually gives you efficient control. So you can imagine having certain systems that are like this, that are basically the only way you're ever going to change the action of that system is physical rewiring. You're not going to convince it of anything. You're not going to punish it into anything. You just have to rewire the hardware bottom up. And then there are some more complex systems like thermostats and so on, where something interesting happens. You can change the action of this very primitive goal-directed system by resetting the set point. You don't have to rewire it. You just change the set point. And in fact, you don't even really have to understand how all of it works. Once you know how to read and write that set point, right? The system takes care of the rest. That really comes into its own with more complex systems that you can train with rewards and punishments. This is why humans have been able to train horses and dogs for thousands of years, knowing zero neuroscience, okay? Because what it does is you're able to, depending on where your system fits on this spectrum, you're able to progressively offload all of the complexity and really onto the system, and really exploit the interface that it's giving you. This thing offers minimal interfaces. This thing offers a little bit, and this thing offers way more. And then, of course, we can, human level communication offers an amazingly rich interface. And so this will become very important, because we're going to talk about what this means for regenerative medicine specifically. This idea that you cannot simply guess where things are. So here's one example. So you look at something like this, and this is a pathway, a gene regulatory network, a protein pathway, something like that. And you might look at this and you might say, well, I can see every element of this. There's no magic here. No, there's nowhere to hide. This thing is the kind of the epitome of a deterministic, reductionist kind of model. Each gene turns another one on and off. That's it, right? That's got to be the level of agency of this thing has got to be zero. That's the standard assumption. So we decided to do an experiment. What if we assume, for the purposes of the experiment, that this thing is somewhere else on this continuum, actually somewhere around here, and we start to treat some of these nodes as inputs as a conditioned stimulus and an unconditioned stimulus and a response. And when you do that, and I don't have the time to go into all of this, but you can look at these two papers, you find out that biological gene regulatory networks, not random networks, only the biological ones, which makes me think that evolution must like this feature. They exhibit 6 different kinds of learning. So this is purely at the dynamical system level. This is without rewiring anything. We're not changing the connections. We're not changing the weights. Simply by virtue of experience, things like presenting multiple stimuli and looking to see whether it learns to associate them. Actually, even this very simple system can do some really interesting, sophisticated things. So what we're seeing is that if you're willing to take this as an experimental kind of a question, you can borrow interesting tools from cognitive and behavioral science to do things in that system that you couldn't do before. And we're now pursuing this, of course, at the bench in biomedical context to deal with things like drug habituation and the fact that pharmaceuticals often stop working after a while and so on. So the fundamental. So let's talk about kind of the scaling of this cognition. All of us take this amazing journey from quote unquote just physics, so the pile of chemicals in an unfertilized oocyte, to eventually something like this, or maybe even something like this. And when we as adult humans will say things like, well, we are different than we are not machines, and we are different than other creatures and so on. We have to keep in mind that we kind of assume that here we are as a finished product, but this process is extremely slow and gradual. There is no developmental biology offers no sharp magical line at which point, boom, before that was just physics, but now we have real cognition. There's no line like that. So we really have to understand how this process happens. It's the most, sort of the most magical. I think developmental biology is kind of the most magical science there is. So what is it that actually scales up here? Well, one thing that we sometimes talk about is the fact that there are these collective intelligences, and people will talk about ant colonies and termites and bird flocks and things as a collective intelligence. And then they will say, well, that may or may not be a collective intelligence, but we are certainly a unified, centralized intelligence. Yeah, we are not like a collection of, like a collection of ants. But of course, if you zoom in, you find out that all intelligence is collective intelligence because we are all made of parts and components. So Rene Descartes was interested in the pineal gland because he said there was only one of them in the brain, unlike other paired structures, and he thought that was very significant. But of course, if you what really is there one of? If you zoom in, you find out that that's full of cells, and inside each one of these cells is all this stuff. Right? So a tremendous amount of components within components within components. And so it's really hard to find anything that's unified in any interesting way. Well, we always have to understand the scaling. So we come from something that roughly approximates this. This is, of course, a free-living organism, but this is a unicellular called a lacrimaria. So these things, you can see there's no brain, there's no nervous system, there are no stem cells, there's no cell-to-cell communication. But what it's very good at is handling all of its local goals in its local at its local scale. So unicellular goals. So all of its morphological needs, its metabolic needs, its physiological needs are all met here in the scale. And the amazing thing is that when these things get together, and individual unicellular organisms get together, they're able to pursue very large scale projects instead of single cell kinds of things. This is the sort of thing they make. They work together to create something very, very large, and we'll talk about that. Now, what does all this have to do with intelligence? So Alan Turing, who probably needs no introduction, was really interested in intelligence, in computing, and in what is common to all intelligence. What is thinking, and how does it exist in the physical world? Interestingly, He also wrote a paper on morphogenesis, on the origin of order in chemical systems that would be important for developmental biology. Now, it might seem kind of strange. Why would somebody who is interested in computation and intelligence be looking at chemical morphogenesis? And I think that he saw a very profound unification. I think he's absolutely right, because I think that fundamentally these are the same problem. These are exactly the same problem, and I think Turing saw that. Here's a simple example. When we look at an embryonic blastoderm, so let's say maybe 50,000 cells of some sort of amniote embryo, you might look at that and you might say, well, there's an embryo. There's one embryo there. And now we ask, what exactly are we counting when we say there's one embryo? Because there are many, many cells, and you might say, well, now what you're counting is the fact that these cells will cooperate towards one particular outcome, meaning the one particular morphological outcome. But what's interesting, and I used to do this work as a grad student, you can take a needle and make little scratches in that blastoderm, and before they heal up, what they're going to do is each region here, because the local activation long range inhibition doesn't cross the scratch, each region will get independently aligned and make its own embryo. And then the thing heals up, and then you end up with conjoined twins or triplets or whatever. So you end up something like this, where there are these regions of embryos and then some cells in between that aren't quite sure who they're part of. But let's just keep in mind that the question of how many embryos there are is not obvious up front. It's certainly not genetically determined. This is a system that can self-organize some number from 0 to probably less than 10 typically, of individuals that you need to count. And so this individuation, this process of becoming an individual from a pool of cells, and how many are going to be there, and who's my neighbor, and where do I end and somebody else begins. This is all right there at the very beginning of development, and it's also there in cognitive science, because if somebody were to show you a human brain and you didn't already know what a human was, and they asked you how many individuals you think live in here, right? How many cells that can this thing house? You would actually have no idea. We don't know. And in fact, in advance, you can't guess that in advance. And we have a very similar issue in terms of you have some sort of medium, and within that medium, you self-organize specific higher level structures, which and much like with embryos, it has a failure mode, right? So we can have split brain patients with different opinions in the two hemispheres about things. We can have all kinds of dissociative disorders and different personalities that live in the same brain, and so on. So this issue of many to one, the scaling is exactly the same across these domains. So here are kind of the main points for today. This is what I'd like to transmit. That if we treat these questions as an engineering problem, we can develop an empirical approach that really tries to find the invariance between very diverse kinds of intelligences. And one way to think about that is the size of the goals that they're capable of pursuing. And I'm going to talk about this towards the end, this cognitive light cone model that tries to capture this notion of the size of the goals that a system can follow. And then towards the end, we're going to talk about synthetic bioengineering and what it means that actually there's an astronomically large latent space of possible bodies and minds outside of the N of 1 example that we see on Earth, and that has huge implications for all kinds of things: evolution, robotics, and ethics, most of all. So the middle part of the talk, what I'm going to do is spend a bunch of time talking about morphogenesis, developmental morphogenesis, as just one example. It's my favorite example, and there are many others, of collective intelligence solving problems in a space, which is anatomical morphospace. So, and I'm going to talk about a mechanism that I think functions as this kind of cognitive glue that binds individual cells into a collective intelligence that solves these kinds of problems. So there'll be four basic sections. We're already started talking about this idea of broadening what we think an agent is, and then we'll talk about the anatomical control, and then some more conceptual stuff, and then I want to show you some synthetic organisms after that. One of the model systems that we deal with is these planarias. These are flatworms. They have many amazing features. One of them is that if you cut off certain regions of the body, let's say the head and the brain, they will regenerate it. And so one of the things that was found by McConnell in the 60s, and then we proved it using modern technology, he was right. We made an automated machine to test this. If you train this planarian, and it's got a centralized brain, and it learns that this little bumpy region here is where it gets fed, you can then amputate the brain. The tail sits there doing nothing for a couple of days, for a week and a half, let's say, and then it grows a new brain. And at that point, you will see evidence that it still remembers the original information. And so now it's very interesting because you see from here that A, learned information can be stored apparently outside of the brain, B, that it can be imprinted onto the new brain tissue as it develops. C, you see this tight integration of behavioral memories and morphogenetic memories, because this thing also has to be able to restructure correctly all of the new cells. And you can also do this interesting, from sort of philosophy 101, this broken transporter kind of experiment, right? With, let's say there's a transporter and you step in it, it transports you somewhere else, but it forgets to delete the original, and now there's two of you. Well, in planaria, you can actually do that experiment. You can cut this thing into many pieces. All of them share the original information, which is the original one, right? So this plasticity, this incredible plasticity, a lot of our concepts start to get really fuzzy. That plasticity is not just for invertebrates. So here is a tadpole of the frog, Xenobos laevis. You will see that there are no eyes up here. We've prevented the normal eyes. Here's the mouth, here are the nostrils, the brain. But what we did do is put an eye, some eye cells, some eye primordia cells on the tail. Not only do these eye primordia cells develop into eyes just fine, even though they're sitting next to muscle and instead of up in the brain where they belong, but it turns out that when they put out their optic nerve, sometimes it finds the spinal cord, sometimes not, but they can see quite well. So we built this machine. This is what we also used to test the planaria. And we find out that we can train them for visual cues. They do quite well in learning to stay out of various kinds of light stimuli and so on. And so you can see the incredible plasticity here. It doesn't need long periods of adaptation to have a completely different sensory motor architecture. So this is, and of course, the plasticity of the brain in terms of kinds of sensory augmentation and things like that. People have done these studies in mammals too. But this just tells you that you don't need long periods of adaptation for this. The system is already poised to accept all kinds of changes to the standard status quo, and that's going to be very important shortly. So how we think about these things is that biology has this multiscale competency architecture that not only are we nested dolls of different levels, right? So organs and tissues and cells and so on, but actually each level solves problems in its own space. It's not structural. It's not only structural. It's actually a functional little problem solver in various spaces. Now, what do I mean by these spaces? Now, one thing that our perceptual systems are very good at is recognizing intelligence in three-dimensional space. So medium-sized objects moving at medium speeds in three-dimensional space, we are very good at detecting agency, detecting intelligent behavior. We know when we're looking at a rock or a mouse or something else. But imagine for a moment if you had an internal sense of your blood chemistry. If you could feel your blood chemistry in all its different aspects, the way that we perceive, let's say, with vision and so on, I think we would then have no problem recognizing that the things that our liver and kidneys and other organs are doing on a daily basis are intelligent behavior in physiological space, in the space of possible physiological states. So there are amazing examples, which we can talk about after. I don't have time to go through them all, of behavior in physiological space, behavior in transcriptional space. So walking around this high dimensional manifold of all the different gene expression patterns that a cell could have. And my favorite, which is morphospace, the space of possible anatomical configurations of a given organism. And one way to think about this is that evolution reuses some of the same tricks, some of the same navigation tricks, which people who work on autonomous vehicles and robotics use exactly the same tricks, to solve problems in different spaces. And from the simplest organisms all the way up to behavioral and then even linguistic in humans, it basically pivots some of the same tricks and the same mechanisms, in fact, to navigate all these different spaces. So what I want to do now is drill down into some examples in one particular kind of space, which is anatomical morphospace. So here's what the problem looks like. Here's how we start life. So a collection of blastomeres. This is a cross-section through a human torso. So look at all the amazing, all the tissues, the organs, everything is in the right place, the right size, with the right neighbors pointed in the right direction. Where does this order come from? And the typical easy answer is, well, it's in the DNA. And of course, we can read genomes now. We know what's in the DNA. It isn't any of this. What's in the DNA is directly is the specification of the micro level hardware that every cell gets to have, the proteins. And using that hardware, all of these cells undergo a physiological process that reliably generates this order. So what we would like to understand is how does this group of cells know what to make? If this a part is missing, how do we convince these cells to rebuild? How do we make them recreate complex structures for regenerative medicine purpose? And as engineers, and I'll show you this towards the end of the talk, as engineers, we'd like to know what else can they build? The exact same cells. What can we get them? What can we get them to build? And so, in thinking about the end game of this whole field, when can we go home? I think it's useful to think about something like this as a target to shoot for, a kind of a design of challenge. What we need is an anatomical compiler. An anatomical compiler is something that future people will be able to sit down in front of the computer and draw the plant or animal that they want, whatever it looks like. It may be a normal organ, it may be something completely crazy and alien, but they should be able to draw any organ, any structure that they want. And if we knew what we were doing, the system would compile that description into a set of stimuli that would have to be given to cells to get them to build exactly that, right? Anything. So complete control. Now, crucial to note, the anatomical compiler is not a 3D printer. The point is not to micromanage the cells and put them exactly where you want. The point is to communicate to the collective the large-scale structure that they need to build exactly the way that this happens under normal standard conditions. Now, why do we need such a thing? Well, among other things, because every problem in biomedicine, with the exception of infectious disease, so everything else, birth defects, traumatic injury, cancer, aging, degenerative disease, all of this would be solved if we understood how to get collections of cells to go to a particular region of anatomical morphospace to make the right kinds of organs with the right shape. We have almost no clue how to do that. Now, there's been amazing progress in genetics and molecular biology. So why don't we already have that? Well, here's this very simple example. This is a baby axolotl, and baby axolotls have legs. This is a larva of a frog, Xenobot laevis. These tadpoles don't have legs at these stages. And one of the things we do in our lab to try to understand the scaling of this morphological outcomes is we make frogolotls. So a frogolotl is part axolotl, part frog. And now I ask a very simple question. You've got the genome for the axolotl, you've got the genome for the frog, full genomic sequences. Can you tell me if a frogolotl is going to have legs or not? And we have basically no frameworks whatsoever to answer this question. We just have a very poor understanding of how large-scale collective decisions are made by components whose molecular biology we do understand quite well. So where we are is this: the field is very good at manipulating this kind of information. We're actually a very long way from doing what we really want, which is complete control over form and function. And I would like to argue that I think where biomedicine today is where computer science was in the 40s and 50s. This is what programming looked like back then. You physically had to interact with the hardware. That should sound very familiar. All of today's most exciting molecular medicine is CRISPR, genomic editing, pathway rewiring, protein engineering, protein structure, all focused at the hardware. Right. And the fact that, all my students laugh when I say, how come you don't program your laptop with a soldering iron today? And this idea that we've learned to take advantage of the software layers. We've learned to take advantage of a more powerful, higher level interface. You don't have to do everything at the hardware level. And this is what we are trying to do in my group. So one of the specific things that we want to take advantage of in these systems is their intelligence. This is what I think we can really capitalize on. What do I mean by intelligence? I like William James's definition, which is the ability to reach the same goal by different means. That's it. He's not talking about how much brain you have or what kind of brain you have. This is a nice, very generic definition that admits of a whole spectrum from extremely simple to extremely sophisticated. So I want to show you some examples of this kind of intelligence. So I don't mean human level. I don't mean linguistic. I mean, different degrees of the ability to reach the same goal by different means when you are interfered with. So here are some examples of what morphogenesis can do. This is the most obvious one. It's regeneration. So this is an axolotl, a Mexican salamander, regenerates its eyes, its jaws, its legs, many other organs. If you amputate anywhere along the limb, these cells rapidly grow and do all kinds of things that get you back to the correct shape. If you keep cutting fingers, it will keep regenerating. No individual cell knows what a finger is or how many it's supposed to have, but the collective absolutely does. And you know that because you can try to deviate it, you can try to mess with the structure, and they will always expend energy to try to get back to that same region of morphospace. And when does it stop? It stops when it has succeeded. It stops when it has made a correct salamander limb. By the way, worms and amphibians are not the only ones that do this. Humans regenerate their livers. Deer, large adult mammal, regenerates tons of bone and innervation and vasculature every year, right, in these antlers. And even human children can regrow their fingertips. So this is a very generic kind of capacity. So one thing is that you're able to reliably, when you are deviated, let's say surgically or by other means, you're able to go back to that. You're able to navigate back to the correct region of morphospace. Now, the path that you take is not necessarily the same one that you tread earlier. So, for example, the way that frog limbs develop originally is you get this flat paddle, and then you kill off a bunch of cells in between the fingers, and now you've got this nice limb. In a story I'm going to show you in a minute, when we induced leg regeneration in frogs, which normally don't regenerate their legs, what we found is that they don't do this at all. They grow like this. There's a central kind of stalk, and off of that will come fingers and a toenail. The most distal part is already here. It's much more like a plant grows. It's not like this at all. It's a completely different path through morphospace that nevertheless gets you to the same final outcome. This is still early, the leg's still growing, but look at these distal elements. I mean, very nice leg. So you can get to where you need to go despite perturbations. You can do it via novel paths, and this is maybe my favorite example ever. This is kind of remarkable. This was discovered in the 1940s. This is a cross section through a kidney tubule in the newt. And normally there are 8 to 10 cells that get together and they build this thing. One thing you can do is you can make these polyploid newts, which have multiple copies of the whole genome, which is amazing that they survive all that, but the cells do. They get larger. The cells get larger and larger. And when you have these, let's say 4N newts, the cells get quite big, and there's fewer of them that still make exactly the same type of tubules, same radius. The animal's the same size, even though the cells are all bigger. So that's kind of amazing. The scaling so the cells are fine having extra copies of the genome. They adjust to having the wrong size, and then when you make, I believe this was 8N newts, the cells get so gigantic that there's not room for even more than one. And what happens then is one cell wraps around itself like this, giving you the exact same kind of lumen. Now, why do I think this is so amazing? Because this is a completely different molecular mechanism. This is cell-to-cell communication and tubulogenesis. This is cytoskeletal bending. So this is a nice example of top-down control. In the service of a large-scale anatomical structure, different molecular mechanisms get called up to implement under novel circumstances. That very much reminds us of certain certain cognitive systems where you have some sort of executive control that tells the animal where they would like to go, and that trickles down to all of the low-level mechanisms, including depolarizing a bunch of muscle cells and everything else that is required to implement that high-level goal. So already we're seeing, and all of this is discussed in kind of painful detail in these papers if you want to see all the details. So what we're seeing again is this idea, James's idea of being able to navigate that space, in this case anatomical morphospace, in novel ways to make up for various blocks that we throw up in their path. Here's a final kind of example to motivate the next part, which is this. These tadpoles are supposed to become frogs. To become a frog, they have to rearrange their face. So the jaws have to move, the nostrils have to move, the eyes, everything has to move. The traditional kind of view of this was that this was a hardwired set of movements, that every organ just goes in the right direction, the right amount, and then you get from a normal tadpole to a normal frog. So we decided to test that. Is it really that hardwired, or is there more intelligence here? So what we did was we produced what we call these Picasso tadpoles. We just scrambled everything. The eyes are on the side of the head, the jaws are up here. Everything is scrambled. And what we found is that those animals make largely normal frogs, because what they basically do is all the components move around in novel paths. Sometimes they even go too far and have to double back, but they move in novel paths to end up where they need to go. So what the genetics gives us is not a hardwired set of tissue rearrangements. What it actually specifies is a system that can do some kind of error minimization. It can deal with unexpected starting positions and move whatever way it needs to still end up in the right place very differently. So the way we've been thinking about this is this standard story that we're told in developmental biology, which looks mostly like a feed-forward open loop kind of system where genes interact with each other. They make some proteins. The proteins act under the laws of physics in parallel, process of emergence and complexity, and boom, out comes something complicated. And of course, that does happen. We know that simple rules can give rise to complex things, but it's far more than just feed-forward emergence. Because if you try to deviate this process, and that might be injuries, it might be mutation, it might be physiological change, whatever, so these systems will activate both at the level of physics and at the level of genetics, all kinds of feedback loops to try to get back to where they need to be, to navigate that space to get to the correct pattern. Now, on the one hand, this is not surprising. Biologists, of course, are very comfortable with feedback loops and homeostatic circuits and all that. But there are a couple of interesting things here that are different. First of all, unlike in a homeostat for, let's say, a temperature or blood pH or the metabolic state or something like that, the set point of this thermostat, of this homeostasis, is not one number. It's an anatomical descriptor. So it's a rich data structure. It's not a scalar. That's first. Second, this process has a real flavor of goal directedness, which a lot of people, especially in molecular biology, don't like. But this isn't magic goal directedness. This isn't a second order, I know what my goal is. It's not that. It's a very simple kind of cybernetic control theory sense of goals. The system will expend energy to minimize error. And the third thing is that it makes a very strong prediction. The prediction is this: if this weird way of thinking about things is true, if it really stores some kind of a set point, then what we can do is make changes by learning to read and write that set point. We don't have to make changes down here. The current paradigm, let's say CRISPR and genomic editing, says that, well, that all the changes have to be made roughly down here. And that requires you to be able to invert this process. That process is generally not invertible. If I tell you that I want a threefold symmetry instead of bilateral symmetry of this axolotl, what genes would you need to edit? I mean, there's no way to reverse this in general. It's a really terrible inverse problem. But if there is in fact a set point, then what we can think is that, well, maybe we can rewrite that set point and let the cells do what they do best, which is cooperate together to build to the set point. And so this is what we've been looking for some number of years, and I'm going to show you exactly how this works. And so so then the question was, okay, well, what's a precedent for this? How come, we're saying that living tissue is going to store a geometric descriptor as a set point? When does that ever happen? Well, of course, it happens in the brain, because we can store patterns, geometric patterns, as goals in brains. So let's say mazes and things like that. And so here's, I don't have to tell this audience what's going on here. We have the hardware, which are these ion channels, and they can propagate electrical state across the network through gap junctions to their neighbors. And then that underlies all of the software that we see that drives behavior. And then there's this process of neural decoding, where the commitment of neuroscience is that all of the cognitive content is here in this physiology. That if we understood the electrical signaling, and this is a zebrafish video that this group made in live fish, that if we understood what was how to decode this, we would have access to the actual thoughts, preferences, memories, behavioral repertoire of these animals. So the amazing thing, and so this is an incredible architecture, of course, that underlies all of this, all of the non-genetic learning and so on. But what's cool is that evolution actually discovered this long before brains showed up on the scene. So right around the time of bacterial biofilms, evolution figured out that electrical networks are amazing at storing information, processing information, integrating information across space and time. And all cells in your body have ion channels. Most of them have gap junctions. And we can do a very parallel kind of research program on neural decoding on non-neural embryonic tissues to ask, can we decode what information is being processed by this network to allow the thing to navigate morphospace, to get from a single egg to whatever it's going to be, frog, human, whatever. And so it's really very isomorphic, where what the electrical network in the brain does is control muscles to move you through three-dimensional space. That same system, much more ancient system, is controlling all of the cells to move the configuration of the body through morphospace. It's a very similar kind of a way to use this network, except the output is different and the time scale is different, right? Because we're not talking milliseconds, we're talking hours, and in some cases days. So the time scale is different, and some things are swapped spatial for temporal patterns, but basically it's the same set of tricks. And so neurons and modern synapses basically speed optimized a lot of stuff that was going on long before that, and you can read all that here. So we developed some tools to be able to manipulate, to read, decode, and write these pattern memories. So first was the use of voltage-sensitive fluorescent dyes. These are slow dyes, slow response dyes. So we're not looking for a millisecond spiking. We're looking at long-term stable resting potential states. And so this is a time-lapse movie of an early frog embryo made by Danny Adams, where you can start to see all the electrical conversations that cells have with each other. Then we do a lot of computational modeling to understand how the various channels and pumps in these cells actually give rise to all of this. I'm going to show you two patterns. This is, again, a video made by Danny of an early frog embryo putting its face together. And again, this is a voltage dye, so the color represents voltage states. And this is one frame taken from that video. What you see is that before the genes come on and before the anatomy is formed, all of the regionalizations of the face, the eye, the mouth, the placodes, already this is what tells all these cells what they're going to do and what the face is going to look like. As I'm going to show you in a minute, this pattern is instructive. If you move any of the states, not the cells, but the electric states of this pattern, the gene expression follows, the anatomy follows that. So this is a native pattern that is required for normal craniofacial development. This is a pathological pattern. If we inject an oncogene, even before the tumor becomes histologically apparent, you will see that the cells acquire an aberrant voltage potential. They start to detach from the electrical network, and they basically treat the rest of the body as just external environment at that point. And we'll talk about that momentarily. So that's how we track these things. Now, more important than that are the functional tools. So how do we rewrite these patterns? And basically, the key thing is there are no fields, no waves, no magnets, no electromagnetic radiation, no electrodes, none of that. What we do is we play on the native electrical interface that cells expose to each other to manipulate their behavior. Basically, we steal everything from the field of neuroscience. So all of the tools, the conceptual tools, but also the practical tools, optogenetics, neurotransmitter, drugs, ion channel mutants, gap junctional pharmacology, all of that stuff works perfectly well outside of the brain. Those tools, they don't distinguish between brains and body tissues. And so we use all the same tools. We manipulate gap junctions, we open and close ion channels with light, with drugs, all the same familiar tools. And so now what happens? Why is this important? How do we know that these electrical patterns are actually functional? So here's one simple example. Here's a frog embryo, and what we've done is we've taken some potassium channel RNA, and we've injected it into a group of cells that are eventually going to become gut. So these are endoderm fated cells. So we inject some potassium channels, and what you see, and what that does is it sets up a little spot of bioelectric pattern, exactly like that eye spot that I showed you in that electric face movie. And so what happens as a result of that is that it triggers eye formation anywhere else. And by the way, outside of the standard anterior neurectoderm, which was thought to be the only region competent to make eyes in vertebrates, actually you can make eyes anywhere. Make them on the tail, you can make them out of gut, prospective gut tissue. so and if you section those eyes, they can have all the same retina, lens, optic nerve. They got all the same structures. So a couple of interesting things here. One is the amount of information we put in is actually quite simple. We don't specify everything needed to make an eye. There's a lot of complexity inside the eye. We don't know how to make an eye. We don't provide that information. It's extremely modular. What we've provided is basically a subroutine call that says make an eye here. The signal that triggers this is a particular spatially distributed pattern of resting potentials, same as for the face, same as for other things I'm going to show you. That's the first interesting thing. The second interesting thing is that here, this is a lens induced out in the tail of a tadpole somewhere. The blue cells labeled with beta-galactosidase are the ones that we injected with our channel. All of this stuff here that participates in making this nice round lens was never injected with anything. So what happens is this is very similar to the kind of recruitment that you see in ant colonies and other collective systems, where these cells, there's not enough of them to make a proper lens. So we instruct them to make an eye. They instruct their neighbors and basically recruit a bunch of normal neighbors to participate in this process of eye morphogenesis. And so this is one of those competencies that these cells have. Not only do they understand a very simple signal to trigger towards a complex path through morphospace towards eye development, they can also recruit as needed to along the way to get the job done. Right? We can make, using the same process, we can make ectopic brains, we can make ectopic legs, we can make ectopic hearts, and otocysts, which are inner ear sort of balance organs. We can also make fins. Now that's kind of weird because tadpoles aren't supposed to have fins. It's more of a zebrafishy sort of thing, and I'll talk more about that momentarily. Of course, this idea of being able to trigger large-scale morphogenesis with a simple trigger is very appealing for regenerative medicine. We're doing this for limb regeneration now. So as I said before, in frogs, normally you amputate. 45 days later, at this stage, there's nothing. What we can do is trigger a particular bioelectrical state, which kickstarts the whole cascade. So immediately you get this MSX1 regenerative positive blastema. Things start to grow right away. In the latest paper from last year, we show that a one-day exposure to this ionophore cocktail drives 18 months of leg growth. So basically, for a year and a half, this on a full-on adult frog, the leg grows. We don't touch it during that time. The only thing we do is interact with that wound right at the beginning for 24 hours to push it towards that path in morphospace instead of scarring leg regeneration. So I have to do a disclosure here because Dave Kaplan and I are co-founders of this company called Morphoceuticals, and we're trying to push that technology delivered with a wearable bioreactor to mammals, so mice and rats at this point, and then eventually, hopefully someday, patients. So I want to switch gears now and really get into this idea of this bioelectric pattern as a set point for morphogenesis. I want to go back to this animal, these planaria. The planaria, of course, are amazing because they're immortal. They are incredibly regenerative. You can cut them into many pieces. And I want to show you one particular story, which was driven by Fallon Durant in my group. And this is so here's your normal one-headed worm. You can see that the anterior genes are in the head where they belong. And if you amputate, that middle fragment very reliably gives you a one-headed worm. Now, here's a one-headed worm. Again, anterior genes in the front, not in the back, where they're supposed to be. But this guy gives rise, when we cut them, gives rise to a two-headed worm. Now, this isn't Photoshop. These are real two-headed animals. Now, why might that be? I just told you it's very reliable. Well, that's because at one point, Wendy Bean in my group discovered a bioelectric circuit that actually sets up a pattern that tells this fragment how many heads a correct planarian is supposed to have. And what we can do, so here's this pattern, one head, one tail. What we can do, and this is still quite messy, the technology is still being worked out, but what we can do is we can respecify this with a temporary exposure to an ion channel drug. We can respecify it, and now we can say, no, two heads. Okay. Now here's the most important part of this whole business. This bioelectric pattern is not a readout of this animal. This bioelectric readout is a scan of this one-headed, anatomically and transcriptionally normal animal. This means that a normal body of a planarian can store one of two possible, and I'm sure there's more, but we've nailed down two possible representations of what a correct planarian looks like. So remember the whole premise of this part of the talk. If the collective intelligence of these cells are going to try to navigate morphospace towards a specific set point, we need to be able to find the encoding of that set point. We need to be able to interpret it, and we need to be able to rewrite it. So this is it. You're looking at it when we ask the question, what determines how many heads a planarian is supposed to have? This is the pattern. Now that's the first thing. So we're looking now at the encoding of the set point. The second thing that's interesting is that this is a primitive counterfactual memory. This is not the map of what's going on right now. This is a map of what might happen in the future if the animal gets injured. And until it gets injured, it doesn't do anything. It's a latent memory. It just sits there until you cut the animal. Then this pattern becomes relevant. Then the cells obey and make a two-headed worm. So this is perhaps, I throw out this hypothesis that this could be a very simple precursor of the amazing capability of brains to do this kind of mental time travel when you can remember things that are not happening now and imagine future things that are also not happening right now. The ability to store this different representation is a very basal version of that. And why do I keep calling this thing a memory? Well, because around 2010, Nestor Oviedo, who's on this call, did some amazing studies to ask what happens to these two-headed worms under future rounds of regeneration. So we take this two-headed animal, we remove the primary head, we remove this ectopic secondary head, we take the middle fragment, no genetic change has been made, we didn't edit the genome, we didn't put any transgenes. Standard genetics, you would think that it would just then go back to normal and make a one head. But that's not what happens. These middle fragments continue to regenerate as two-headed in perpetuity. Here they are. There's a video of taken by Junji Murakuma about what these guys are doing. As of a few years after that, we figured out how to set them back to one-headed, so we can take the two-headed form and reprogram them. So this has all the this electrical circuit has all the properties of memory. It's long-term stable, it's rewritable, it has conditional recall, and it has discrete possible behaviors. Notice then that the question of how many heads do planaria have is not really nailed down by the genome. What the genome does is specify an electrical circuit that, left to its own devices, reliably generates a pattern that says one head, but it's rewritable. It's the actual information is stored at the software level, rewritable by just 48 to 72 hours of exposure to a stimulus in physiological space. So part of the lab now is focused on this, basically pulling together what we know about the state space of the electrical circuit with current ideas in connectionist thought about how electrical networks can store information, recover from errors, and things like that. So this is kind of the computational work that we're doing. Now it's interesting. It goes beyond mere head number. It goes also towards head shape. So this animal with a triangular head can be treated in a way that just for a couple of days screws up the ability of the cells to regain the correct electrical pattern. And then you can get flat heads like a P. felina. You can get round heads like an S. mediterranea. In fact, not just the head shape, but the shape of the brain inside and the distribution of stem cells becomes exactly like these other creatures. Again, no genetic change, but you can access other species morphologies that are 100 to 150 million years evolutionary distance. So what the electric circuit can do is select between attractors in its state space that correspond to other species. And you can start to think that that's a very quick way of making that change. And then evolutionarily, it could perhaps through Baldwin effects and assimilation, it could eventually, maybe that's how these species came to be in the first place. We don't know. But you can also access regions of that state space that look nothing like modern planaria. So you can make these crazy spiky forms, you can make things that aren't flat at all, and you can make combinations. So what we really would like to do is to have a full stack kind of understanding, starting with the molecular genetics of all the channels and pumps that are expressed, followed by using this amazing simulator that Alexis Pytak has made, large-scale electrical gradients that exist in the field, so the electric phase and these patterns and so on. And then from there to the logic and actually the symbolic logic that underlies how these types of patterns make decisions about how many heads, where the heads are going to be, and so on. And then we can apply effective interventions. I'll show you a very quick way to make use of something like this. We started to look at so therapeutics, birth defects. We started to look at one way that brains get malformed. And so there's all kinds of teratogens, nicotine, alcohol, all kinds of drugs and mutations that will screw up the early brain. So what we did was we made a computational model of the electrical pattern that tells the early brain what size and what shape to be, okay? And then we asked what happens to this pattern when it's hit by different drugs. And I'm running out of time, so I'm not going to go into details unless people ask afterwards. But basically, what you can see, this is the what I think is the most kind of impressive example. So here's a normal brain: forebrain, midbrain, hindbrain. Here's one with a notch mutation. So this has a dominant overactive notch. So you can see the forebrain is basically missing. The midbrain and hindbrain are a bubble. These animals severely defective. They have no behavior, they're just extremely impaired. So what we did was we asked the model, the computational model, a simple question: what channels could we open or close to get from the incorrect bioelectrical pattern, which is one of the things that this notch mutation does, is it actually screws up the electrical pattern. What could we open or close to get back to a correct pattern? The model gave us one idea, which was the HCN2 channel. And what we found is that if you then either open that channel with drugs or simply inject new HCN2 channels, despite the fact that you have this notch mutation, you get back to normal. So the shape of the brain, the gene expression in the brain, and the IQ of these animals becomes indistinguishable from controls. So this is very interesting. While I'm not claiming that this is always going to work this way, there are many things that are not going to be fixable this way. This is an example of fixing a hardware defect, an actual mutation in an important neurogenesis gene in software, by providing temporary electrical states to the brain that reset the morphogenetic traversal of the system. And so this is, of course, these things are also now being tested for various kinds of birth defects. And so down the line, we want this system, which is going to let people design these electroceuticals. So at some point, and you can play with a kind of an early version of this. At some point, you will take whatever the incorrect pattern is, and it might be a birth defect, it might be a wound injury, it might be a tumor, and you can use the system to ask, first of all, what kinds of channels are even in there. That's just transcriptomic and proteomic data. But then we can use the simulator to ask what kinds of ion flows would get you back to normal, and then choose, now that you've picked your tissue, you know what the channel and pumps are, so you know what your candidates are, and then you choose from known drugs that target all of this stuff, and there's a massive toolkit of ion channel drugs already out there. Which ones of them would get you what you need? So it's a design platform for generalizing the frog brain story I just told you. So in the last 5 minutes, I'm running way behind. So what I'm going to do is I'm going to skip all this until unless people want to talk about this at the end, and then I'll come back to it. And I just want to the slides, in case we want to come back, are all about actually coming back to this idea of scaling this navigational intelligence from single cells, and some hypotheses about how that happens. But what I want to make sure we get to is this idea of novel organisms. So one of the things that we were interested in is to understand how much plasticity exists in cells that normally have a very reliable kind of behavior in developmental biology. And if we did make novel creatures, what would their morphology be like? What would their behaviors be like? And where would these come from? And we got an amazing opportunity to do that using a substrate of frog skin cells. And this was all done in collaboration with Josh Bongard's lab, and Doug Blackiston in my group is a staff scientist who did all the biology, and Sam Kriegman in Josh's lab did all the computer science for this. So what we did was this. We took an early frog embryo. Here it is in cross section. These cells up here they're animal cap cells. They're going to make ectoderm, so skin. And what Doug did was basically dissociate. So here they are. We dissociate all these cells, and we put them in a little depression all by themselves. So we're not adding. There are no transgenes here. There's no any kind of nanomaterials. We're just basically taking them away from the influence of the other cells. And so what could happen? Well, many things could happen. The cells could die. They could crawl away from each other. They could do nothing. They could form a two-dimensional monolayer like cell culture. Instead, what they do is they come together overnight. They sort of coalesce, and they make this interesting little thing, which we call a Xenobot, because Xenopus laevis is the name of the frog. So one of the things it does is it repurposes the little hairs that it has on its skin. So frog skin has little hairs that rotate to move the mucus down the body of the frog. And so these are now repurposed to actually swim, to row against the water. And you can see that in the particles here. They can go in circles. They can sort of patrol back and forth like this. You can make all kinds of weird shapes. So here Doug made kind of a donut that moves along. And they also have all kinds of collective behaviors. So you can see some behavior tracking here. Here, these two are interacting with each other. This one's going on some sort of long journey. These guys are doing nothing. Here's one navigating a maze. So it goes up here. It's going to take this corner without bumping into the opposite wall. So it takes the corner, and then here spontaneously it turns around and goes back where it came from. So some sort of internal dynamics are causing it to change direction at some point. Okay. They have regenerative capacity. So if we cut them in half, they will. So here we cut them in half. There's this kind of hinge here, and they will go ahead and reseal themselves into their new Xenobot shape. If you look at the calcium signaling here, they have very interesting dynamics, which we can apply all the tricks from neuroscience, right? So mutual information, transfer entropy, all these kinds of things to find out, is it brain-like? To what extent are they communicating with each other? We don't know. I make no claims about that at the moment. We're just starting that. But keep in mind, there's no neurons here. So all this calcium signaling here is done by the skin cells. And all the behaviors you've seen are done purely by skin. And so now in the last couple of slides, I want to show you one more amazing thing that they can do. So by taking this skin biobot away from the rest of the cells, we've made it impossible for these guys to reproduce in the normal frog-like fashion. But if you provide them with loose skin cells, so these white things are just more Xenopus cells, what you find is that they move around both individually and in collectives, and they make little piles. And what they do is over time, they make these little piles and they push the skin cells into little balls, and they sort of compact them. And because much like us and much like evolution, they are dealing with an agential material, meaning that these are not passive particles. These cells, when they come together, they know how to do morphogenesis. Because of that, what happens is you get this reproductive cycle, which we call kinematic replication. These little balls that they make mature overnight to become the next generation of Xenobots. And guess what they do? They run around and collect more, and they make the next generation. And that makes the next generation. So this is basically von Neumann's dream of a robot that goes around, finds materials, and makes copies of itself. Now, this is extremely primitive. There is no strong heredity here. So we have to provide them with these loose cells, so they don't have the ability to sort of generate them on their own. So it's a very primitive example, but it's kinematic replication, and this happens within 48 hours of liberating the cells. To our knowledge, no other animal on Earth does that. So this raises some interesting questions. What does the Xenopus laevis genome actually encode? Well, most of the time it's a series of standard developmental stages and then some tadpole behaviors. But actually also this is an early Xenobot. This is a month-old Xenobot. It has some kind of developmental stage here. I don't know what the heck this is. This is some kind of novel thing, and they have different behaviors. So this is interesting. A couple of things to think about. One is that we didn't engineer in this case. Of course, we will in the future add synthetic circuits and whatnot, but we didn't do any of that. All we did was liberate these cells from the influence of the other cells. So if you think about what does skin know how to do, the fact that it has this boring two-dimensional life on the outside of the animal, keeping out the bacteria, that's what it does when the other cells behavior shape it into doing that. On its own, left to its own devices, this is the lifestyle that it has. And you would never know that by watching standard development. So standard development hides a lot of these weird competencies from us because it's so reliable. And that's the first thing. The second thing is, for any animal, if you ask typically why does it look the way it looks, why does it have certain behaviors, the answer is, well, because for eons selection has shaped it to be this and that. there's never been any Xenobots. There's never been evolutionary pressure to be a good Xenobot. So the question of where do these capacities come from is really interesting. And we don't know, by the way, we're just now beginning the kind of cognitive experiments. Can they learn? What kind of conditioning can they do? Do they have preferences? What problems can they solve? We don't know. We have some hints, but I'm not making any claims about that yet. Okay, so I'm just going to finish up here by pointing out this. Because biology is so incredibly good at organizing something coherent in novel circumstances, any combination of some sort of evolved material, designed material, so nanomaterials, smart chips, whatever, and AI, is some combination of it is going to be a possible viable agent. So we're talking about cyborgs, hybrids, instrumentized organoids, chimeras, all of these things, so all of the natural forms that Darwin called endless forms most beautiful, these things that really impress him with their wide variety, all of that is this tiny corner of possible space of beings. And so this really begins now to push us towards the need for some new ethical platforms, because I think that in the next few years, we are going to be surrounded by creatures who don't share an evolutionary lineage with us, because their structure will be completely different from anything that we can point to on the tree of life, and they're not going to share an origin story, because they will be partly evolved, partly designed. So we need new ways to interact with each other, with other beings with potentially diverse types of intelligence that do not rely on what you look like and where you came from, right? That origin story, these criteria that we've used for a really long time of, you sort of walk up and you knock on this new system, and if you hear a metallic clangy sound, you say, okay, that, you come from a factory, you're probably pretty boring, I could take you apart, it's all good. And if it's soft and furry, then you treat it a different way. Those kinds of things are not going to survive the next couple of decades. We need a completely new framework for understanding this. I'll just end here by saying that all of this, there are these are some of the more conceptual papers. If people are interested, I'm happy to send this stuff out. I want to thank the postdocs who did, and the grad students who did all of the work. We have some amazing collaborators that work with us, some amazing technical support. The model systems really do all the heavy lifting for all of this stuff. I thank our funders, and that's it. I'll stop here and thank you for listening, and I'll take questions.